Good evening, everybody. I'm Michael Klonsas, and together with my colleague Pinar Yilmaz, I'm happy to welcome you to this USOMI webinar. Today, we have the great pleasure to have with us Dr. Andrey Fedorov, who is an associate professor of radiology at the Surgical Planning Laboratory at the Department of Radiology of Brigham and Women's Hospital and Harvard Medical School. Dr. Fedorov joined the Surgical Planning Laboratory in 2009 after obtaining his PhD in computer science from the College of William and Mary in Virginia. His research is in translation and validation of medical image computing technology in clinical research applications, with a focus on quantitative imaging, imaging informatics, and image-guided interventional procedures. Dr. Fedorov is committed to advancing the role of reproducible science, data sharing, and open source software in academic research. He has contributed to a number of open source projects, most notably the one that you may all know, 3D Slicer. And together with Dr. Kikinis, he is currently a co-principal investigator of the team tasked with building the NCI Imaging Data Commons. And today's talk will be exactly about that, the NCI Imaging Data Commons towards fair imaging data via standardization and openness for AI. Before we start, I would like to tell you that you should write your questions down in the Q&A box, and we'll discuss all the questions together in the end of the talk. And for now, Dr. Fedorov, the floor is yours.
Thank you very much for the introduction, and thank you for the opportunity to present our work at this venue. And thank you all for joining this seminar. Here are my disclosures. We are funded by the US federal government, and the views that I present are mine and do not represent the views of the US government. So here's the team that is behind Imaging Data Commons. I want to start by acknowledging the team, which includes a variety of expertise, and without this team, Imaging Data Commons would not happen. So next, I want to talk a little bit about the motivation, what led to the effort to establish Imaging Data Commons. And to a large degree, the motivation was established centuries ago, where the basic premise for scientific work was established, that if you want to prove that your method works, it's your responsibility to make it so clear and transparent how you achieved the result. Otherwise, nothing can be taken for granted. And I guess every generation has their own challenges how to meet the demands of reproducibility. And we have our own share in this age of artificial intelligence. For a very long time, scientific manuscript was that evidence that was well established as the artifact that accompanies every scientific study. But as we work our way through the new challenges in AI, it is more and more recognized that availability of data, software, and to a large degree hardware is critical for establishing reproducible science. Unfortunately, meeting these challenges is not easy, and very often you find papers that do not meet all of those challenges, not because they're willingly not, because the authors do not know exactly how to approach these complicated tasks. If you look at the data, imaging data in medicine is very complex. It covers different modalities, resolutions, domains. We are dealing with patient information, so identification is of absolute importance. Sizes of data, when you go to large clinical trials, when you look at microscopy data, sizes can get very large. And it takes significant effort to curate this data. And furthermore, it's important not just to share the original imaging data, but to establish an environment that allows for incremental enrichment of data, meaning that if I share the images that I collected in my study, I want other people to be able to analyze and share analysis results of this data, or, for example, annotate this data. And as you all know, for AI research, availability of data is of absolute importance. Some are saying that in the future, the data will define, will supersede the source code and will be equally or even more important. And of course, the quality of data is defining the effort that will need to curate it for specific applications. And studies show that major effort goes into curation of data. A lot more effort goes there compared to developing new AI methods, for example. Sharing software also is very challenging. We have more and more dependencies that are maintained by different groups that are continuously evolving, and packaging those dependencies, making sure that someone else can run your software is not easy, even though more and more tools are becoming available to facilitate the process of sharing. And another I guess, complication is that once the software is shared, to some degree, it's up to the authors to maintain the software, to answer the question, to fix bugs, to upgrade it as the library's dependencies are changing, and that requires continuous effort. However, without sharing the software, it's becoming more and more recognized that the results presented in manuscripts may not only be misleading, but actually harmful, because it was well established that very prominent studies sometimes are not possible to reproduce. And going forward, both the funders and publishers are demanding more and more accompanying the publication manuscript with the software and with the ways for reviewers and readers to reproduce the findings presented in the paper. Finally, hardware is a relatively new component that contributes to the complications in establishing reproducible science. The GPUs are evolving continuously. There are many configurations that you can choose from. Very often, those configurations affect performance of your algorithm. And very often those choices that you need to make about how many GPUs you want to use, what are the memory, what is the driver that you installing on your machine, can have significant implications on your result. And because these resources are sometimes sparse, sometimes are very expensive, it becomes even more complicated for the reader to reproduce the study that you are presenting. So these challenges have been recognized at different levels, and in the United States, in the final years of the Obama administration, Joe Biden pioneered the Cancer Moonshot effort, which aimed to reduce the barriers and accelerate the search for treatment of cancer. As part of establishing the specific goals of Cancer Moonshot, a scientific panel was established that defined main priorities for what we should do as the scientific community to achieve the broad overall goals of the Cancer Moonshot. And one of those specific tasks that was defined was to establish a national cancer data ecosystem. And that panel, that scientific panel that defined those priorities, recognized that sharing and integrating data, providing easy access to the data within the community will be very important for accelerating cancer research. And right now, the infrastructure to enable this is not there. And that is why the cancer data ecosystem was recommended for to be funded and to be created. Going a bit down, if we want to achieve this, if we want to establish this infrastructure for allowing people to access and share the data and enabling analysis of data, what are the guiding principles? I think the first guiding principle is we would like to have the data that is fair in this infrastructure. And fair, findable, interoperable, accessible, reusable is a concept that was introduced at this point a few years ago, and it defined the requirements that data should meet in order to be recognized as fair. And it is very important that the FAIR principles put specific emphasis on allowing the machines to interact with the data. It's not what you as a user should put in the documentation for the data set, but it's more how can you enable APIs and software to search the data, because that's what you ultimately need to integrate data across different groups, across domains, and truly make the data interoperable. And the second guiding concept in building this national cancer ecosystem is the concept of data commons. And the data commons is a concept introduced by Bob Grossman, also around the same time as the concept of fair data, that suggests that with the emerging capabilities of the cloud computing, instead of performing the research in a traditional way, where every researcher or every center have their own computing infrastructure, you have your laptop, you have your cluster at your institution, and traditionally, the idea is that you take the data that is located in the in an accessible data repository, download the data to the location where you have the computing infrastructure, and do your work locally. So what are the problem here? With this approach, you inevitably replicate the data, you introduce inconsistencies in the data, while it's very manageable to maintain computing infrastructure locally, it becomes very challenging to reproduce the infrastructure that you have, an environment that you configured by other users. So sharing and reproducibility is becoming challenging when you do research in a traditional way. Furthermore, if you at some point become constrained at the computing capabilities, for example, your laptop doesn't have the GPU that you need to accomplish specific tasks, it becomes difficult to scale up and grow your infrastructure. So what was suggested by Bob Grossman is that with the cloud computing, instead of bringing the data to the local computational resources, you utilize the cloud that provides elastic access to network bandwidth, computing infrastructure, disk space, and you collocate your tools and you configure your environment in the cloud. If you this approach makes a lot of tasks a lot easier compared to the traditional way, because if you want to share the environment where you performed your experiment with someone else, it's just a matter of modifying the security configuration in a specific machine, or providing the scripts that will allow another user configure the resource in the same way that you did. Cloudway provides a lot of other approaches, because arguably it can simplify the security implementation. It clearly can scale a lot more easier than what you could do locally. And also, you only pay for the resources that you actually use. As soon as you are done with your research, you can shut down the virtual machine, and you don't need this big investment in order to get started with the analysis. So these are the two principles that are behind the NCI Cancer Research Data Commons, which is that framework that is being set based on the guidelines by the Scientific Board of Cancer Moonshot. And Cancer Research Data Commons, or CRDC, is a cloud-based data science infrastructure focused on cancer research that includes the repositories of data that are domain-specific, but also combines a variety of tools that allow users to analyze this data, explore this data, visualize it, and includes both the publicly available data sets and also those that are restricted access, and access control to those data sets is provided by the standard API. CRDC has very ambitious objectives to support cancer research. It's intended to cover a broad range of domains, imaging, but also genomics, proteomics, preclinical data, a variety of capabilities, and primarily CRDC is targeted to support big data collection efforts that are funded by the US National Cancer Institute, such as the Cancer Genome Atlas, Human Tumor Atlas Network, and such. So these programs generate a very large amount of data, and collection of this data is supported by taxpayers' money through the grants. And it's very important that there is a place where this data can be archived and accessed by the community. So Imaging Data Commons, in turn, is just one of the components within broader CRDC. And Imaging Data Commons, IDC, is a cloud-based repository of publicly available cancer imaging data co-located with the analysis and exploration tools and resources. So it's very important to understand the difference. The CRDC is everything within this red circle, and IDC is one of the data repositories within CRDC. So let's talk a little bit about the definition of IDC and what this all means. So first of all, IDC is a cloud-based repository. And what this means is that all of the data we have in IDC is located in the cloud. Currently, we use Google Cloud Platform as the architectural foundation for IDC. Going forward, we are also looking at replicating IDC data on Amazon infrastructure to make it more accessible to the broad range of users. Why do we do this? Is because by co-locating data in the cloud, we facilitate the use of the data commons concept. We facilitate access to the data from cloud-based tools and computational resources. At the same time, if for any reason you are not interested or not familiar with the cloud and you would prefer to analyze the data locally, this is still fine. You can access the data we curate in the cloud, and you can download it to your computer, to your cluster, and proceed with the analysis as usual. We would like to encourage you to explore the cloud approach to analyzing data, but you definitely don't have to do that. What is inside Imaging Data Commons? We are tasked with curating cancer imaging data across domains and across different applications. We started populating IDC with the data from radiology, but over time we already have a very large amount of digital pathology data, H&E, microscopy slides, and more recently we added fluorescence imaging data from the Human Tumor Atlas network that contains dozens of different channels. And going forward, as we identify new interesting data sets that are relevant and important for cancer research, we are planning to curate those as well. Very importantly, IDC is not just about images, but also about image-derived data. So we already have a number of annotations, volume to locate the data in the clouds to make it easier to use tools and resources that allow you to use the data. And there are different categories of those tools. Google provides a number of big data tools that allow you to effectively and efficiently query the data, provision computational resources, develop interactive dashboards that allow you to search the data, and many others components, some of which I will describe in my slides. But in addition to the generic tools available from Google, there are also tools within CRDC, so-called cloud resources that are focused on scientific workflow and are designed to work with the data repositories of CRDC. And of course, we want any tool developed by a cancer imaging researcher to be usable with our data. And towards that end, we are developing example notebooks, example workflows that demonstrate how to take those community developed tools and apply them to our data. IDC development is funded by a contract from Leidos Biomedical Research, and we were funded in the summer 2019. We are aiming to follow agile development process, and because of that, very quickly after the award, we released initial pilot version of the platform that had a relatively small amount of data, but allowed to demonstrate the basic concepts of what we are trying to achieve in order to seek feedback from the users. Our initial goal was to ingest radiology data from the Cancer Imaging Archive and curate that data in the cloud. We proceeded with the production release in the fall of the last year. And as of today, we have more than 40 terabytes of imaging data. All of it is in public collections, and primarily it's radiology and digital pathology coming from a variety of sources, not just TCIA. And here's a brief summary of in numbers what we have right now in IDC. And this is the front-end portal for IDC that should serve as a gateway for getting started with our resource. Here's another view at how we evolved in terms of the number of files and total data size. So every dot here corresponds to a data release. And you can see that the process was by far non-linear. In this case, for example, we had big jump because we ingested national land screening trial collection that alone had about 10 terabytes of data. And at this moment, we are aiming to have regular releases every month or so, where we will synchronize all updates from the data coordinating centers we are working with and making those available in the cloud and to our users. Here's another view at the current snapshot of the data that we have in IDC. As you can see, we have a range of manufacturers, different modalities that are covered, and a broad range of cancer types. So the idea is that if you develop an algorithm, for example, that appears to work well on the data collected at your institution or at the consortium you're working with, you can use, for example, sample images from IDC to test the robustness of your algorithm on a variety of samples of data and manufacturers. So the data that we have available is important, but I think more important is what we enable you to do with the data, because data alone without the tools and without the ability to explore the data is potentially not very useful. So the overall idea of what we want to achieve in IDC, we want you to enable to do as much as possible with the data without having to download anything to your computer. You start with imaging data commons from the IDC portal that I'm showing here in this interactive figure. From this portal, you can navigate to the exploration page, and in the exploration page, you can see a subset of metadata attributes that allows you to select data sets of interest. You can select those data sets, you can view them, and you can view both the images and annotations, as is shown here with the segmentation of kidney and kidney tumor in this example. But the portal is just the first step in starting to explore what is available in IDC. We have other interfaces, most importantly, SQL interface that allows you to query and subset the data in a very detailed fashion by writing SQL queries and defining persistent cohorts of data that you can reference, for example, from your publication. So if we think back to the definition of the FAIR principles, in imaging data commons, we leverage the ability of the machines to interrogate all of this data. The collections we have in IDC come from a variety of centers, groups, and equipment types. And in order to be able to achieve the uniform interface to searching the data, we need to have consistent model and consistent representation of metadata. You can think about imaging data commons as a library. And in the library, if you want to find something, but you don't have the catalog, the library becomes pretty much useless. In imaging data commons, the metadata that accompanies images, accompanies the files that we are curating, is that catalog that allows you to understand what is available in the library and identify the subsets that are of your interest. In order to achieve this, in order to harmonize metadata and provide this uniform data model, we rely on DICOM, Digital Imaging and Communications in Medicine standard, which is a well-established standard that has been around for decades, and up to recently it was mostly used in the clinical world. But as we are discovering over time, it's a very powerful standard that can provide a lot of utility in research as well. So why DICOM? First of all, it's a standard that has been around for a very long time, and it's well established in the imaging field. And to the credit of the imaging field, it's hard to find similar standards in other fields, such as genomics, for example. It's a standard that is well accepted and is used virtually in every radiology department in the world, and right now is also becoming accepted in the digital pathology field. DICOM allows us to harmonize data across domains, across data centers, vendors, applications. And very importantly, it allows us to prepare for the future. Because of the breadth and the power of the standard, we have pretty good confidence that future data types, future capabilities, and future needs of the community will be met by this standard. By using DICOM standard, we don't need to invent a new standard that will allow to interoperate for the commercial and open source tools. It's the standard is already well implemented in the community. And if we look at the FAIR guiding principles that are defined by this paper, we have, I would argue, complete coverage of those principles between what is available in DICOM standard and the infrastructure that is implemented in Imaging Data Commons. If you go one by one here, by design, when you create a DICOM object, it will automatically be assigned unique and persistent identifier. It will always be accompanied by rich metadata. When you create image-derived objects, such as segmentation, you have a way to refer to the images that you segmented. So by doing this, you provide the provenance of your data and you link different data components together. And with the IDC platform features, we provide infrastructure that allows you to leverage the richness and power of DICOM metadata. And we provide you with the tools that allow you to search and interact with this data. So you may be well aware that not all data in imaging research originally is in DICOM representation. Very often when we talk about radiomics features, annotations, segmentations, those come from research tools that are not DICOM aware. And because of that, the process of ingestion of data into IDC includes harmonization of data into this common representation. And this is a significant effort, but we believe that this effort is well justified because it allows us to provide uniform interface to the data. I want to also emphasize that Imaging Data Commons hosts data that has previously been de-identified, and we take identification and patient privacy very seriously. It is not the role that IDC serves itself to de-identify the data. This is done by the data coordination centers that collect the data. This could be the Cancer Imaging Archive or other centers we've been working with. And it is up to that center to establish identification policy and implement that policy to remove all patient-sensitive information from the data. Once the data has been identified, we have close collaboration with data coordination centers to understand their de-identification procedures. Once we see that those meet the requirements of identification, we accept the data and then release it in our cloud-based infrastructure. So this is the process that we are working on right now for receiving imaging data. Going forward, for accepting derived data. For example, if you work with images already available in IDC and you annotate those images with segmentations of tumors, we are considering developing a process where those annotations could be ingested into IDC directly without having to go through the data coordination center. So going back to DICOM, why we implement this, it's not just about FAIR data and organizing the metadata. DICOM has a lot of capabilities that are very critical for operating a resource. For example, DICOM defines what are the attributes that should be scrubbed in the process of de-identification. So we don't need to come up with a new standard for identification. We can follow established standard that has been reviewed by the community and that is implemented in a variety of tools. DICOM provides means to integrate with other standards. And very importantly, something that is not recognized often in the research community, DICOM is an evolving standard. In the process of curating data in IDC, we often face situations where we see some gaps or in the standard or where clarifications are needed. And we are working with the standard community, with the working groups that work on putting together the standard to refine and improve it. Finally, very importantly, DICOM also defines the web services, the interfaces that allow you to query the data and retrieve data over the web. And we leverage those DICOM web interfaces very heavily in IDC. As I said before, we want you to be able to do with the data as much as possible without having to download it. And visualization is this basic capability that we provide, because you need visualization in order to be able to assess the quality of the images, in order to establish that whether there are artifacts in the images and such, and also to see the annotations that accompany the images. So we, within IDC, provide implementation of the web-based viewers that allow you to look at the images. And when you open the image, the URL that you have in your browser is persistent. So for example, if you found something interesting, you can copy this URL and send it to your colleague. You can use it from your notebook. So it's you can bookmark this image, for example. We think that this is a very important capability that we implement. And we do this for radiology data and also for the digital pathology data. As you can see here, this is an example where we display it's a pathology image in the browser. As you can see, the speed is quite good. So if we continue to this more complicated example, this is an example of a fluorescence image that was contributed by Human Tumor Atlas network. And in this case, we have quite a number of different channels, and you can interactively choose which channels you want to see. You can adjust the opacity and colors. And it's very important to emphasize here that data representation is the same using DICOM, and the access to the data is achieved again through the same standard DICOM web interface. So we do not need to do any customization to support this new type of data. The standard is powerful enough to meet these needs. Well, another component that is very critical for us in order to implement image and data commons is the Google Cloud Platform. So this is the suite of cloud computing services that are very robust and scale elastically based on the needs. We leverage Google Cloud Platform to maintain storage of the files that we curate, maintain metadata in BigQuery tables that you can search using SQL. If you want to analyze the data in IDC using Google Cloud Platform, you have access to highly scalable compute resources, a variety of GPUs, and specifically targeting AI. There are hosted notebooks, Colab notebooks that you can access to define reproducible workflows, and AI notebooks if you want to have a configuration that leverages powerful GPUs, and if you want to have high flexibility in resources that are available to you. One of the components within Google Cloud Platform that is critical for IDC is the Google Cloud Healthcare API. So this API implements the concept of DICOM Store, which essentially is a hosted DICOM server solution. It's a DICOM server solution that is effectively maintenance-free. You can create this DICOM server with a click of a button. You do not need to worry about patching that server to address the next security vulnerability. You do not need to worry about scaling your capabilities of your server if, for example, you need more disk space or if you need more RAM. All of those things are taken care behind the scenes by Google, and you have a convenient interface to populate this server with DICOM data and retrieve this data through the standard DICOM web interface. So as part of our workflow, we import our files into cloud storage buckets. From storage buckets, they go into the DICOM server. And once they are in DICOM server, there is again another click of a button functionality that allows you to extract DICOM metadata into standard tables accessible through SQL. The healthcare API also provides identification capabilities. We are not leveraging those in IDC because the data we receive is already identified, but a number of projects are leveraging this capability, and it's quite interesting if you want to look into this. So on the Google side, we have a proprietary commercial implementation of the DICOM web interface that makes those images available through the web. On the browser side, we leverage open source tools that implement the same DICOM web interface and talk to the commercial solution to retrieve the images and show them to you in the browser. The viewers that we integrate with IDC are open source. For radiology image data, we use OHIF viewer that perhaps many of you heard about. And for the digital pathology, we leverage the SLIM viewer developed by Marcus Herman. Both of those, again, are open source. And as we identify bugs or missing features in those viewers and we contribute those back, those also become publicly available. So what you see in imaging data commons when it comes to visualization can also be easily reproduced outside of IDC. Okay, I talked about BigQuery before. I will just mention this, that this functionality is very, very powerful if you want to search large amounts of data. As I mentioned, we have more than 40 terabytes of data. And with 40 terabytes of data, it becomes not feasible to download the entire data set to your computer and then select files of interest on your own. Of course, it's feasible, but it will take a very long time, a large disk, and it's not clear why you would want to do that. With Google BigQuery, we simplify the task of identifying relevant data within IDC. So, for example, if you look at the NLST collection that has about 10 terabytes of data, and you have a question, I want to identify all series that have slice thickness less than 1 millimeter. If you do it in a traditional way, you would need to download all of the files. You would need to extract DICOM metadata. You would need to set up a viewer in order to examine the quality of the data. So this will, I would be surprised if this takes you less than a few hours. With imaging data commons, you don't need to download anything. You write a simple SQL query. You define the characteristics of the data subset that you're interested in. You can run this query within seconds, and you can visualize the images within seconds to confirm that what you found is actually what you need. And then once you identify the files that are of your interest, you can download just those files. And the query is quite simple. You can see here that basically this is the public SQL table, BigQuery table that contains all of the DICOM metadata. You say that I want to see all studies and I want to have the GCS URLs. These are the locations on the storage buckets where the corresponding files are located. And then here you specify the criteria for search. I want to look at only CT scans from the NLST collection that have slice thickness less than 1 millimeter. And again, if you want to download the data, it's okay to do it either in the cloud VM or on your computer. We have this covered in the documentation here. So I would argue that IDC provides really unprecedented capabilities thanks to the DICOM standard and Google Cloud Platform curation of the data for visualizing and subsetting this large amount of publicly available cancer imaging data. But we don't stop there. We don't stop at allowing you to search and explore the data. Once you identify the subset of data that is of interest to you, you can define those persistent subsets of data. You can define the cohorts using the unique identifiers. So if you publish a paper and you used a subset of data for training your model, you can include in a supplement to your paper exact list of studies that you utilized in training, and your reviewers can have access to that same data set to confirm that your algorithm works. We provide examples how to integrate the data with the cloud-based tools. We have many examples: how to build interactive dashboards using Data Studio, how to develop interactive workflows using Google Colab, how to query the data using BigQuery. So all of those tools are available to you at your fingertips to work with IDC data. If we go back to the motivation of developing data commons and IDC, I believe that with the resources we put in place, it's becoming a lot easier to establish reproducible computational workflows, because you can freeze essential computational environment, you can define the characteristics of your VM, you can freeze the input data and output data, you can have those in persistent location, and the tools that are available from the cloud help you achieve this task in a relatively straightforward way. So the primary tool that I always emphasize is the Google Colab. So Colab is a hosted environment that provides you a Jupyter-like interface to write a code and accompanied by a text to explain what you are doing. Google provides this to you for free and will give you a free virtual machine that can have a GPU to run this code. And once you developed your notebook, you can share it with your colleagues. You can include a pointer in your publication. And within this notebook, you can access the data that we curate in Imaging Data Commons. So you start, for example, with a notebook source that is available in GitHub. You launch a Google Colab VM instance. You have cells that install the prerequisites on this VM. You fetch the sample or cohort from Imaging Data Commons. And by going through the analysis steps, you can create your annotations, your segmentations, you can develop visualizations. So you have a complete life cycle of the workflow that links to persistent locations of code, prerequisites, and source and output data. And of course, Google Colab is free, which means there are limitations on what you can do with Colab. There are limitations on the type of GPU, amount of memory and disk, but you can also connect Colab Notebook to a custom compute engine virtual machine. So you can configure virtual machines that are as powerful as you want or as you can afford, and use the same interface to Colab to run those workflows. One of the most common questions we get with the cloud is how much does it cost? And the reality is that when you use advanced capabilities of the cloud, sometimes it's difficult to estimate the costs. And that's why Colab is perhaps a good first step towards exploring the cloud, because you can have very tight control over what you spend. You have the subscription of $10 per month, and it's very affordable and in our experience already very powerful. And I want to emphasize that Imaging Data Commons is not sponsored by Google. I'm not trying to promote this product. I don't have any conflict of interest. I'm just mentioning this as it's a very valuable scientific tool in our opinion, based on our experience. So if you go to Imaging Data Commons documentation, you will find examples of notebooks that go through the step-by-step configuring the environment, installing the community-developed AI models, fetching the data, providing you with the visualization of the results. So I think best is to come up to your own conclusions and look at the documentation examples we have developed. Finally, very often you want to utilize non-imaging data in your analysis. And I started by saying that Imaging Data Commons is just one of the data nodes available within CRDC. There are other nodes that contain proteomic, preclinical, genomic data. And within CRDC, there are tools that allow you to find matching data for your imaging cohort. So integration of imaging with non-imaging data is another capability that hopefully will be valuable to our users. So going forward, we definitely have a number of challenges. I talked about fair data and DICOM, but the reality is that the effort to establish a fair data, create fair data, and work with standardized data is often not insignificant. We have a lot of work to be done to simplify those processes, to develop documentation and tutorials to help users interact with our data. And the concept of using the cloud for the research also is quite novel and relatively unexplored. For the past several years, we've been working on developing use cases that utilize Google Cloud for real scientific analysis, and we are in the final stages of preparing publications that will demonstrate how this can be done, accompanied by reproducible notebooks and data cohorts. But definitely there's a lot of work to be done to better understand what are the use cases where cloud is beneficial and where we should stay with the on-premises resources. Going forward, we are working to ingest more data into IDC, but also at the same time, we are working to enrich the data that we already have. So we are very interested in creating annotations for the imaging collections that we are hosting, and we are making steps towards that. We are working to ingest clinical data into IDC so that you can subset imaging cohorts based on clinical characteristics of your population, not just image acquisition attributes. User outreach is very important to us. We have tutorials and in-person and webinars. And of course, we have a lot more work to do in developing the peer-reviewed publications that demonstrate how IDC can be beneficial to the cancer imaging community. I mentioned before that we are working with Amazon to replicate IDC data in that cloud, because we recognize that a number of users work with Amazon. And we have a lot of work going on integrating imaging data with other components of CRDC to make it easier to run computational workflows that utilize imaging data, and also to integrate imaging data with genomics data, for example. If you want to get started with IDC, please check out our website. We also have a publication that introduces main concepts behind IDC. And although you can do a lot without any cloud credits, we also provide, we have a program where we will award you pilot allocation. So you can utilize this pilot allocation for any non-free resources. For example, if you want to allocate a more powerful virtual machine, or if you want to deploy your own DICOM server. So if you go to our homepage, you will find link to documentation and instructions how to apply for IDC pilot credits. I also recognize that it's a lot of stuff that I talked about today, and I didn't have time for any demonstrations. If any of what I said was interesting to you, please join IDC community office hours. So we have those every week, twice a week, to accommodate different time zones. So you can, it's a free form meeting. You can come with any questions, and usually I'm there or someone else is there from our team, and we can give you demonstrations and explain in more detail how to navigate IDC and how to benefit from the examples that we developed. So finally, I want to acknowledge the technical team behind Imaging Data Commons. Thank you for your attention, and I hope you will explore Imaging Data Commons on your own after this talk.
Thank you very much, Dr. Fedorov. This was amazing, a wonderful overview, a lot of information.
Thank you.
For the first presentation after our summer break. And there are a lot of questions coming in as well. So I'll just to start with the questions.
I can see the Q&A panel. If you like, I can just read them and answer if it's okay.
Yeah, I would want to read them and you can answer.
Yeah, so I see there is a question from Eleftherios Trivisakis,
yes.
Which is basically, is there any provision for integrating clinical data in the most standardized way into the IDC portal? Typically, clinical data are provided as an Excel spreadsheet, making it challenging to track those down or to identify novel clinical endpoints. It's very timely that you're asking this question. Thank you for asking it, because we are in the final stages of working on release 11 of IDC. And in release 11, we will have clinical data ingested from a variety of Excel spreadsheets and CSV files. And in V11, we will have that clinical data available as BigQuery tables. I want to emphasize we are not aiming to harmonize the data across different collections, but more we want to provide a single interface that you can use to search the data. So instead of going to specific collection page, download Excel spreadsheet and understanding what is there, you can have one single table where you can run your queries, and we hope that it will be easier to understand. But we would be very interested to have feedback from users like you. Please join office hours or send a message on our user forum. We will have example documentation how to access clinical data. Hopefully it will be live this week. So stay tuned.
Amazing. I can imagine how much work that is, because I mean, nationally, we have a backlog, I know from certain research projects, also within the Rotterdam study, in getting cancer registered data, which has to be double checked, of course, before using. I was also wondering, in how, is it already clear data that you can all implement in the IDC, or how does that work?
Right. So we are not as ambitious as you in the goals of making clinical. We are not taking data from cancer registries like nationwide cancer registries, but more we look at the collections that we already have, imaging collections that we already have in IDC. And some of those, when they were submitted by the submitters, they were accompanied by clinical data spreadsheets. And quite a few actually were, probably more than half have some clinical data. And up until now, that clinical data existed somewhere in the spreadsheets and some files you had to download. So we are just bringing this data into the resource together, so you can search it together with the imaging data. Right. Okay, so
the next one about the DICOMs. I mean, there are two questions.
Yes.
Yeah.
Some DICOM data sets have proprietary text. How this data is handled? Yeah, that's a great question as well. So in DICOM nomenclature, they are called private tags, and these are vendor-specific attributes. Private tags are ingested into BigQuery as well. There is it's not as easy to search those, but they are available in our metadata. If you have specific questions, it's best to initiate discussion on the user forum if you are interested in specific private tag. I think in general, it's hard to give general answer to this, but we have the data. Okay, so next question is, are the DICOM files organized in a PAX or folder structure? So DICOM files are organized in a storage bucket as a flat list right now. There is no folder structure. The idea is that we're not encouraging you to navigate the folders. Instead, we want you to use the BigQuery tables and SQL interface to subset the data and identify the files that meet your search criteria. Once you identify the list of files, then you download just this list of files. And then you can organize them in a hierarchy. And we actually have examples how to do that. So we go away from the folder hierarchies to more data model based hierarchies, because folder hierarchies are not sustainable. And
so it's just a filter. So you can find
essentially.
them with, yes.
Mm-hmm. Right. So it's like this. Go ahead.
No, please go ahead.
You remember I showed you the example of the query that subsets CT images that have certain slice thickness. So that's the concept. That's the idea. And once you have the result of the query, you'll have the list of files that you can then download from the bucket.
And with the DICOMs, I mean, there's a lot of information, hidden information as well. So if we go back with the big data, which comes close also with the GDPR issues, so you have these planes, right, in which you could almost identify certain persons as well. Do you like get rid of them, or How does that work?
Yes. So it's actually significant effort goes into making sure that before the data is released, it does not have any of the metadata that would allow tracking back the patient. So the DICOM standard defines which attributes are potentially can contain patient-sensitive information. And then it also includes rules. What should be done about those attributes? Sometimes they should be removed. Sometimes they should be scrubbed, like modified, for example. We work with data coordination centers, such as the Cancer Imaging Archive, that implements the rules defined by the standard. And then in addition to that, they have a manual review of the attributes. So they actually visually confirm that the values for some of those attributes do not contain PHI. The easiest way to clean up metadata is to get rid of all of the metadata, right? And then you just have the image, but then you are unable to understand where the data comes from, what kind of manufacturer, what are the acquisition parameters. You cannot link images that belong to the same study, same series.
Filter. Yeah.
Right. Yeah. So it's not proper to just get rid of all of the metadata to identify data.
Okay, thank you. The last question in the chat is, to what extent these data can be used other than the validation purpose? It would be much easier. You could give us some example that where and how we should use this data. Also, more importantly, where do you expect us not to use this data?
Well, that's an interesting question. Maybe, oh, sorry, let me.
Because it was interesting what you showed with the collab as well. I was thinking of getting your way through the IDC, and it's very nice that you can actually see what others have done, and that you don't have to, create the wheel again or find your way.
Exactly.
But also to know on which people are working on, or maybe people can collaborate on as well. Is there data on that as well, or some? I mean, you showed something at the end, right? To
yes.
Contact
so
you.
I think there are maybe several dimensions to this question. One is, what can you use specific data for? So every data set we have has the license that accompanies the data set. So you can have, and I think the main distinction between the data set is Creative Commons, which basically doesn't put any restrictions. You just have to acknowledge and recognize the contributor. And then there are some data sets that have NC, non-commercial use. So for example, if you don't want to touch non-commercial use data set, you just select Creative Commons, and you will only see the collections that meet the specific criteria. But what you can do with the data, Creative Commons, you can do anything. You don't need to, there are no constraints. As long as you acknowledge the source of data, you don't need to report what you did. You don't need to ask any permissions. But yes, then the second dimension, what you asked is, do we know how people actually use the data? We don't have a lot of knowledge about that. And we intentionally did not want to create an environment where you had to log in or request access and explain what you plan to do with the data, because that introduces a barrier. Yeah. As you could see, I just went to the website. I clicked this exploration. I don't need to log in. I can build my cohort here. I can view the data. I can download the data. I don't need to ask anyone's permission. So we want to be very inclusive and open. But indeed, we do have the user forum. So if you go from here to discourse, this is the place where we encourage our users to ask questions and share their experience. And sometimes users do have comments about the notebooks that we developed. So for example, this user needed help with the CT nodule segmentation notebook that we shared. Admittedly, we don't have a lot of discussions where users tell us what they do with the data, and it's okay, I think. We would welcome to have those discussions. If you want to ask questions, you're very welcome. So we hope that user forum will help us build that user community over time.
Also, one of the things why I asked this is, for example, the nice thing that you just showed with the segmentations, or that people are rating things or using the data in which they could also give some input in how they've rated certain things, so that it's also adding on the database. I mean, there's also some kind of bias for that as well. I mean, do you do a double check, or I mean, it's open source data, and how reliable is that data then? But that was a thought of, you know, building on the concept of having open big data in when you're using it for, I mean, in developing a tool or using some segmentation or doing some ratings.
Yes, that would be, you know,
the data as well.
Yeah, that's the ideal situation where we have the community that is feeling welcomed to discuss data, discuss the challenges with the data, because I think it is a valuable result to say that we tried this collection and actually it has a lot of data that's useless. Maybe it has a lot of artifacts. You know, that's a very useful information. Or maybe on the other hand is to say that we think the data is valuable. We did a segmentation. Here's a segmentation. Can you include our data in your portal so that others can benefit from it? Because something that is science for me may be just a step towards the science for somebody else. Maybe my research focus is segmentation, but for somebody else it's radiomics extraction using segmentation, or someone it's correlating with genomics, you know. So we are not there yet, but that would be the ideal arrangement where we have this vibrant community of people helping each other.
Yes, and that's why I felt like, is that like some kind of a discussion forum, or that people can exchange ideas, or that you have some kind of, not the real control, because then I totally agree with you, then you lose the purpose of being the open access and not having to log in, not having to control, or actually take responsibility in what people do with the data. But then again, for the next steps in science, I think it's nice to have that, and it's what you do. I mean, it's amazing that you have like these weekly meetings in which you can help people with their questions. I mean, there must be a lot of great discussion going on there. There was also, I think, a question by Michael: are the data backed up by pathology results?
Well, I guess it depends. It depends on the collection. For some collections, you do have the pathology correlates, and that usually is in the clinical data that will be available in IDC. Some collections that we have both the radiology and pathology images. So, for example, if you go to collections such as CPTech, and you look at some of those images, you can find collections that have radiology and pathology. Oh, perhaps this is just pathology. But yeah, I don't have time right now to run the SQL query, but you can define a query that, yeah. So there are different, I guess, ways to answer that question. Clinical data and then actual pathological information with the images.
And you have like a certain documentation, I think, like in help in how to find or define your
we do have documentation, but you know, so it's always a difficult task to write documentation, because you don't, with my perspective, I don't particularly know what are the real user view. So we need this user feedback to say that we want to do this. We looked at documentation. We can't understand what's happening. Can you help us? So that's what we are trying to do with the office hours. Is the users who are confused by the interface, by the documentation, are welcomed to join and ask in person. And then hopefully, based on that feedback, we can refine documentation and improve it.
And for me, you mentioned, I mean, you work with like the Google platform, and you're thinking of Amazon. How did you in the first place end up with Google? Like, why Google?
Yeah, that's also a very common question we are being asked. So there are many reasons, actually. One reason is that when we started developing IDC portal, we based it on another portal that was already in place in CRDC. So we worked with the team at Institute of Systems Biology that used Google for that purpose. They had a lot of expertise, and because of that, we were very confident it would take us much less time to develop it on Google than, for example, on Amazon. Another very important reason is that Google implemented the healthcare API that was focused on DICOM. No other cloud provider at that time, I think still it's the case, had support of DICOM. So that was very helpful to us. Again, we would be able to achieve reliable capabilities in a short period of time. I think these are the two main reasons why Google. But going forward, we are not exclusive. We, you know, as resources allow.
It was just in my, because it's, now it's really clear. I want to thank you a lot for this great presentation and overview, and it's nice that you put this on. So people, if you want to join for using the data, please join the sessions. I guess feedback is generally a big thing within science and going forward in science as well, and especially in big data. So thank you again for this great initiative and your presentation. The recording will be available soon on our YouTube channel and our website, so it's definitely possible to watch this great webinar again. And thank you all again for being here and with us during this webinar. I want to remind you on our annual meeting in October, the 14th and 15th. So please join us in Valencia as well. And then we'll continue with the great webinar series accompanied by Bayer in October as well, the 25th of October. So stay tuned for great webinars. And yes, we do have a YouTube channel, so please go to the OSOMI website. And if you type in OSOMI webinars in YouTube, then you can find the webinars as well. Have a great evening or morning or afternoon.
Thank you very much for inviting me. Thank you.
Thank you very much for accepting, and hopefully see you soon in the future.
Thank you.
Bye, everyone.